Imagine a world where diseases are no longer a threat, where genetic disorders can be cured, and where human capabilities can be enhanced beyond imagination. This may sound like science fiction, but thanks to a groundbreaking technology called CRISPR, this future is becoming increasingly within reach. CRISPR, short for clustered regularly interspaced short palindromic repeats, has emerged as a powerful tool in the field of genetic engineering, holding the potential to revolutionize medicine in ways we could have never imagined before. To understand the impact of CRISPR, we must first grasp the basics of genetic engineering. Genetic engineering involves manipulating the genetic material of an organism to alter its traits or characteristics. This manipulation is achieved by introducing new DNA sequences into the organism's genome or by modifying existing genes. In the past, genetic engineering techniques were limited, costly, and time-consuming, making them impractical for widespread use. However, with the advent of CRISPR, the landscape of genetic engineering has been transformed. CRISPR is essentially a molecular tool that allows scientists to edit DNA with unprecedented precision and efficiency. It utilizes a naturally occurring defense mechanism found in bacteria, which enables them to recognize and destroy viral DNA. This mechanism consists of two key components: a guide RNA molecule that targets a specific DNA sequence and an enzyme called Cas9 that acts as a molecular scissors to cut the DNA at the desired location. Once the DNA is cut, the cell's natural repair machinery comes into play, allowing researchers to introduce changes or insert new genetic material at the site of the cut. The power of CRISPR lies in its simplicity and versatility. Unlike previous genetic engineering techniques, CRISPR is relatively easy to use and can be applied to a wide range of organisms, from bacteria to plants and animals, including humans. This has opened up a world of possibilities for medical research and treatment. One of the most promising applications of CRISPR is in the field of disease treatment and prevention. Genetic diseases, such as cystic fibrosis and sickle cell anemia, are caused by mutations in specific genes. With CRISPR, scientists have the potential to correct these mutations and restore normal gene function. In fact, researchers have already achieved significant success in using CRISPR to treat genetic disorders in animal models. For example, a team of scientists at the University of Pennsylvania used CRISPR to successfully treat a form of blindness called Leber congenital amaurosis in mice. By editing the defective gene responsible for the condition, they were able to restore vision in the animals. This groundbreaking achievement has sparked hope for the development of similar therapies for human patients, potentially offering a cure for previously untreatable genetic diseases. In addition to treating genetic disorders, CRISPR also holds promise for combating infectious diseases. By targeting the DNA of disease-causing pathogens, such as viruses and bacteria, scientists can potentially disable or eliminate these harmful organisms. This approach could revolutionize the way we fight diseases like HIV/AIDS, malaria, and tuberculosis, which have been major global health challenges for decades. Moreover, CRISPR has the potential to enhance human capabilities beyond traditional medical interventions. This concept, often referred to as human enhancement, raises ethical considerations and debates. While the ethical implications of genetic engineering are complex and contentious, it is important to acknowledge the potential benefits that can arise from responsible and thoughtful use of these technologies. One area where human enhancement could have a profound impact is in the field of regenerative medicine. CRISPR could be used to modify the genetic material of stem cells, allowing scientists to engineer tissues and organs for transplantation. This could alleviate the shortage of organ donors and provide personalized solutions for patients in need. For instance, researchers have successfully used CRISPR to edit pig genomes, potentially paving the way for xenotransplantation, the transplantation of pig organs into humans. CRISPR also holds promise for developing new and more effective treatments for cancer. By targeting specific genes involved in cancer development and progression, researchers can potentially disrupt the mechanisms that drive tumor growth. This approach, known as gene therapy, has shown promising results in preclinical studies and is currently being tested in clinical trials. However, while the potential benefits of CRISPR and genetic engineering are exciting, it is crucial to address the ethical considerations associated with these technologies. The ability to modify the genetic material of living organisms raises questions about the limits of intervention and the potential for unintended consequences. One of the primary concerns is the possibility of designer babies, where parents could select specific traits for their children, such as intelligence or physical appearance. This raises questions about the ethics of creating a society where genetic advantages are determined by birth rather than individual effort or merit. There are also concerns about the potential for unintended off-target effects of CRISPR editing. While CRISPR is highly precise, there is still a risk of unintentionally altering genes that are not intended for modification. This could have unforeseen consequences on the health and well-being of individuals and future generations. Furthermore, the accessibility and equitable distribution of CRISPR technology must be considered. As with any groundbreaking technology, there is a risk of creating a divide between those who can afford genetic enhancements and those who cannot. It is essential to ensure that these technologies are available to all, regardless of socioeconomic status, and that their use is guided by responsible regulations and ethical frameworks. In conclusion, CRISPR and genetic engineering have the potential to revolutionize the future of medicine. From curing genetic diseases to enhancing human capabilities, the possibilities are vast and awe-inspiring. However, as we venture into this new frontier, it is crucial to approach these technologies with careful consideration of their ethical implications. With responsible use and thoughtful regulation, CRISPR has the power to shape a future where medicine transcends the limitations of the past, offering new hope and possibilities for all of humanity.